Alright, so now we're going with genetic engineering. We have just finished with mutations, and we have mentioned that mutations are unwanted changes in the nucleotide sequence. Now we have genetic engineering. This is also a way of changing the DNA sequence, but this is for a certain purpose, for a certain good, and usually for the benefit of mankind. And organisms that have undergone genetic engineering, species, so to speak, are known as GMO, or genetically modified organisms. So these genetically modified organisms have certain traits that are desirable for us for nutrition or for economic purposes. Because, for example, root crops or poultry that are genetically modified are larger or have a higher yield, or even are more nutritious or have particular characteristics, such as for fruits, you have some GMOs which have large seeds at the start, or have seeds, and GMO or genetic engineering allows them to be seedless, something like that. So these are things that you want, alright? And this arises from changes in DNA, right? So the name of the DNA, or the description of a DNA which has undergone genetic engineering, is recombinant or chimeric. Why recombinant? Because it is a result of a combination of several genes from several sources. That's why it also had the name chimeric, from the root word chimera. And the chimera is a mythological creature that is a combination of body parts of certain animals or several animals, like the meaning of recombinant, having several sources. Alright? So how do we do genetic engineering in particular? Here we focus on the bacterial DNA, a small bacterial DNA, which I hope you guys know now already. The name is known as a plasmid. In a nutshell, what you do is that you open up a plasmid, you cut it at a particular portion, you put a particular gene, let's draw it in green, you isolate this one, you insert it, and then after that you now have this one, which is the recombinant. DNA. So I'm just going to discuss very briefly how this goes, how this happens. Alright, so first thing, we open up the plasmid. So we have enzymes that cleave phosphodiester bonds. Again, these are nucleic acids, don't forget. And the ones that cleave these ones, alright, or nucleic acids, are obviously known as nucleases. Particularly an endonuclease and a restriction endonuclease. Why did I say restriction? This one means that, alright, goes to say that the endonuclease would only be preferential to a particular sequence. If the endonuclease does not detect that sequence in a particular part of the plasmid, it will not cleave it up, or it will not cut it. Alright? But if it does detect the particular sequence, then it will cut it only at that particular portion. We know that enzymes are specific to a particular site, alright, based on stereochemistry and molecular geometry. For example, we have sequences here at the bottom. For example, the endonuclease would target this particular portion. What would happen is that after cutting it off, it will produce this. Alright? For example naman, here we have this one. The endonuclease would cut it in this manner. After cutting it off, you have portions like this. This one is flat. The ends here are flat. That's why you call this a blunt end, to put it bluntly. Now, this one, it produces, it has a sort of shape, alright, but they call this a sticky end, alright, in comparison to a blunt end. And the sticky ends are the ones that are more common. Now, what happens after you have this, you cut it, then you open it up, it's opened up, then you have this gene, alright, this portion isolated from somewhere, and then you insert it here. And this is the one with a particular characteristic that you like, which you put inside. And then when you have it, you need to seal them off, alright, in order for them to be completely joint, because remember, you cut it off at the start. You need to return it, you stick it back, and since you cut off a phosphodiester bond, you need an enzyme that will seal a phosphodiester bond. Remember replication? Remember the last enzyme that seals off the last phosphodiester bond? That's the same enzyme that would target the closure of the recombinant DNA, and that's a DNA ligase. In the process of opening it up with an endonuclease, inserting a new gene, and then finally sealing it off to form your chimeric DNA is known as transformation. Now the problem here is when you perform this is they say that this is very hard to do, and the problem in addition to that is that you only produce very small amounts. That's why you use bacterial cells. For example, this is a bacterial cell. It has a genome of its own, its nucleic acids exposed in the cytoplasm. What you do, since you have small amounts of recombinant DNA, and one thing that we should know about bacteria, or we should remember about bacteria, is that they divide rapidly. So what you do is you put the recombinant DNA here, and then the DNA would reproduce very quickly. And as a result, you have many copies now of the plasmid owing to the quick reproduction of bacteria. Alright, so that's what they do. However, there are certain cases wherein in a population of bacteria, there are only some bacteria that are able to take up the plasmid. Not all of the bacterial cells could actually accommodate this one. And so how would you be able to trace that? Well, there's a certain part of the plasmid called a selectable marker that will allow you to know whether the bacterial cell actually has the plasmid, which is recombinant, or not. And luckily, one obvious characteristic of selectable markers, or a DNA with selectable markers, is that it allows resistance, or it confers resistance to a particular antibiotic. So, for example, I have here a petri dish, and it has a lot of bacteria, and you don't know what bacterial cells there contain or have taken up the recombinant DNA or not. What you do is you put an antibiotic here, like an antimicrobial plate, alright, and then you would now come up with of course smaller number. You assume that the cells that have died did not get the DNA, or did not take the recombinant DNA, and now the cells that are remaining here, all of the cells that remaining here, they have the recombinant DNA. Alright, so that's it. Now, another thing that you have to know is that there is actually a method wherein you can actually quickly produce a lot of the DNA for purposes of study or for purposes of production. The name of that method is a polymerase chain reaction, or PCR. And you need here a thermocycler. That's what they use, with several other reagents. You want to produce a lot of DNA in a short span of time, and you need the starting materials. What's in a DNA? Of course, you need the nucleotides. Alright? You have DNA, so you need here nucleoside phosphates, I mean. And we need magnesium ions as cofactors for the enzymes in the polymerase chain reaction, and that and that enzyme is known as the Taq polymerase. Of course, you need the solvent, water, which should be nuclease free. Of course, if it has nuclease, then there's no point. You want to produce nucleotides, but it has enzymes that break up the nucleotides, and of course the DNA. This is sort of replication, but what you call this? Sped up replication. And the steps are 3. You want to denature it. Remember, when you replicate the DNA, you first open it up. The double helix becomes opened by helicase. This is what you want to happen here first. This is done at a very high temperature, alright, around 94 to 96 degrees Celsius according to my reference. Then it needs to anneal or cool down after the chain reaction, and this one is at around 55 degrees Celsius to 60 degrees Celsius, and it will extend. Alright, it will extend at around 72 degrees Celsius. So these are the 3 steps in polymerase chain reaction. You denature it, then it simmers down, and then that's the time that it will expand to produce the multiple nucleotide chains. And so that's it in a nutshell for genetic engineering.